Great. So welcome everybody to today's seminar. My name is Petra and I work at TATAA Biocenter. Now TATAA is a Swedish company. We have a base in Gothenburg. And what we do is for almost 20 years now, we have been organizing molecular biology training, both theoretical and hands-on. And today education is actually a quite small part of TATAA. And what we do now is that we provide molecular biology reagents and consumables. Some of this is our own production, mostly qPCR and quality control tools. But then we represent a number of different companies, different manufacturers that we trust and that we know that they deliver a good quality product. So for example, Swift Biosciences and VarSome that today's webinar will focus on. And what kind of sets us apart from other companies that represent different manufacturers locally is that we also have a service lab, and we are participating in many different research projects as well. So we actually use these products that are not manufactured by us, but what we distribute. So we can actually share our own feedback of these products, and then we can also help our customers, for example, in NGS, for those who do not wish to prep libraries themselves, they can send samples to our lab and we can prep libraries and do Illumina sequencing in-house. And what we do is we offer a complete workflow, anything from sample collection and extraction to, for example, the library prep kits and the data analysis tools that will be today's focus. So this is just a number of different instrumentation that we have available in-house. And that's it for the short introduction of what TATAA does. And I will hand over for Sharadas. You can share your screen.
Yes, thank you very much, Petra, for the introduction. And I'm going to share my screen in a minute. So can you see my screen now?
Yes.
Perfect. Yes, good afternoon, everyone. Again, my name is Gerardus. I work for Swift Biosciences. Our distributor in Sweden and Norway is TATAA, and we work with them. They are our representatives on our kits for that market. Without further ado, I would like to start mainly focusing today on our oncology and custom amplicon panels, which is used for low frequency variant detection. And this is in conjunction with VarSome Clinical, who will extend our presentation today. We work with them over a year now. So the quick snapshot of the company. Swift Biosciences develops a NGS library kits that are compatible for Illumina and Ion Torrent platform. We have developed a broad portfolio of IP to protect our unique chemistries that are used in the library construction. We focus on the cell-free DNA, FFPE samples. We also have the kits for the PCR-free libraries that will allow you to do whole genome sequencing, targeted sequencing, amplicon sequencing, bisulfite sequencing, and to do custom amplicon as well. In addition, we also provide the kit that I will mention briefly today for the library normalization prior pooling that takes away the workflow of using the qPCR in your workflow. The company is based in Ann Arbor in Michigan, and it's also ISO certified. This is our portfolio in more detail, and today I'm going just to focus on the targeted sequencing in our Accel-Amplicon panels. And we just recently launched SNAP panels, which is Swift Normalized Amplicon panels. This is for normalization of your libraries prior pooling. So the workflow, you can see here, it's a 2-hour protocol, basically enriching your target with the multiplex PCR and adding then the adapters by ligation, index adapters, and then this is streamlined into the sequencing. And the data that got out of the sequencing can be analyzed. And more and more we are getting customers that use VarSome Clinical that Thomas will go into more detail. A few highlights why we are different from the competition. It's a single tube assay. It's a short protocol. You can achieve the library in 2 hours. And it's a low input. We accept the sample down to 10 nanograms of cell-free or FFPE DNA. And as I said, it works on Illumina and ion-turned platforms. Most of the panels limit of detection is 1 or higher in terms of allele frequency, so higher than 1%. We also design our panels so we cover all the reads you get back are most likely to be 90% on target, and the coverage uniformity around 9%. And this is compatible with cell-free DNA, so with low inputs. As I said, it also supports limited amount of DNA, and it's a cost-effective approach for your enrichment by Amplicon strategies. So this is how we compare to our what's out there on the market. It's a short protocol. It's also a single tube protocol, and you can do the assay in less than 2 hours. So this is the available panels that we have on the shelf. As you can see here, our main seller, we have high volume of panels of 50 for our customers for 56, which are called 56 or 57G. These are pan-cancer pre-validated panels that you can buy off the shelf. This is just to give you a bit more detail of what the panel consists of, which genes we target, and which variants of clinical significance are covered, indicated here. And this is how they split. And for example, here we have variants that you could see here are pathogenic, regarded or referenced in the literature, and there are about over 1,000 of them. And this is all based on the American College of Medical Genetics or American College of Molecular Pathology guidelines. This is our latest edition, 57G pan-cancer profiling panel. You can see here, it's the uniformity of the performance of the panel and in different inputs indicated here. I would like to highlight the cell-free DNA. That's how it performs. So you get 95% on target in dark blue columns, and coverage uniformity, again, 95% in this wider blue columns, bars. As I said, this is the 10 nanograms of input. It's a pre-validated content with 16,000 cosmic and clear VAR mutations found in more than 20 common cancers. And as I mentioned, this panel is a streamlined analysis through VarSome. I also wanted to highlight that not only we cover the hotspot mutations indicated here with low frequency, we also cover the deletion, deletion implicated in non-small cell lung carcinoma, and it's highly referenced now in literature that responds to the drug therapy. Apart from the off-shelf panels, we do have the custom panels. And this is the process how we design the custom panels. So the customer provides us the gene coordinates. We come up with the design, we send the bed file, you load the bed file, and then you could see the coverage of the exons or the regions of your interest. Once you are satisfied with the design, we finalize it, and within 4 weeks, you get the panel on your doorstep. It's important to highlight that Swift conducts wet lab testing on the control DNA. So we do a shallow or low coverage sequencing, making sure that your panel performs to our standards. And then after the delivery, we also can work with you to provide the best support, and hopefully get you in your routine projects on using Swift custom design panels. So this is how the IGV browser shows the exon that is covered with the Swift amplicons. So this is what we would provide you upon your request to design the custom panel. Now, just wanted to briefly highlight what we, how we are referenced in the literature. So this is one of the selected publications from 2019 molecular case studies. This is the correlation or the comparison of NGS results with the validated with digital PCR using our 56G oncology panel. And what I wanted to highlight is the allele frequencies that we have found using the cell-free DNA using NGS, so 0.6%, and then it was validated with digital PCR here, which found the allelic frequency of about 1.9%. So this is highly correlated. And also looking at another gene, for example, T53, using sequencing and then validating this by digital PCR. So just to highlight that our panel gives high correlation between NGS and the digital PCR. So we just very recently and very briefly wanted to highlight the new version of the Amplicon panels. So this is the old protocol, and we launched recently called SNAP, Swift Normalase Amplicon panels. So these are the panel, this is the workflow that is very similar to our previous workflow, but what is included is the normalization step when pooling your samples prior sequencing. So you're achieving a very uniform coverage of the across the samples in when you're sequencing them. So this is just a brief graphic view of how the Normalase works. So your libraries here, indicated here, your Amplicon libraries. It's a Normalase PCR, which is a part of the indexing PCR. So it comes from there. This is a part of your library prep. And then it's a Normalase, which would select a certain fraction of your library to be maintained, to be protected in the final pool, which is Normalase 2. And then Normalase 2 gets rid of the excess of the libraries in your final pool indicated here. And you end up with equimolar concentration of each of your sample in your library pool. So this is the actual data from University College London here. These are not Amplicon, these are exome sequencing data, but what I wanted to show, this is the principle of the Normalase, how it works. So you get a very uniform sample coverage across your samples, so you're not over sequencing most of the samples and not under sequencing. And the coefficient of variation between the those libraries is around 3.71%, which is unachievable by doing the qPCR quantitation of each library and then pooling based on the qPCR result indicated here. So this is the libraries that were by qPCR, and the coefficient of variation was around 20%, and using the Normalase, it's 5.6%. So you have a very even coverage, a very even stacked coverage of your libraries. So with this in mind, I would like to thank you and turn over to Thomas, who will talk about the variant detection. Thank you.
All right, Gerardas and Petra, thank you for introductions. Now I'll take over and I'll give you a presentation about our company and services for interpretation of NGS data. So let me share my screen. So now I think my presentation is going to take about half an hour, 30 minutes, and during my presentation, feel free to paste your questions in either in the chat window or in the section for Q&A. And we will take these questions and answer them at the end of our joint presentation. So I'd like to start with a quick introduction to our company. So, okay, so my name is Tomas Kusera, and I'm the Director of Business Development at Safeter. Safeter, it's a precision medicine and bioinformatics company. We are headquartered in Switzerland, in Lausanne. We are on the market since 2014, almost 6 years now, and we develop tools for processing and interpretation of NGS data, specifically for clinical purposes, although we also serve research customers in certain cases. From our perspective as a precision medicine company and bioinformatics company, we see a number of challenges in bringing NGS technology to the clinical practice. There are many challenges for sure, but from our perspective, we are focusing on 3 challenges in particular, which are all related to the interpretation part of the pipeline. So the challenge number 1 we are facing here is the fact that the annotation data are fragmented and spread over many data resources, right? So it's a very time-consuming process to look up a specific annotation data in all these fragmented, data resources. On top of it, our understanding gets better pretty much every day, which means that what was valid yesterday doesn't have to be valid tomorrow. So we have to constantly keep an eye on the latest discoveries in order, to increase the diagnostic yield over time for NGS-based testing. And the third challenge we are facing here is the lack or a little standardization in terms of interpretation of pathogenicity, meaning there might be 2 laboratories interpreting the same variant very differently as a consequence of having access to different annotation data resources. So in total, the interpretation part or the interpretation of pathogenicity, it's a very, very complex endeavor and very time consuming. And we are focusing on these 3 challenges in particular. So these 3 challenges are precisely the reason why we have created VarSome. I'm sure some of you have seen it before. VarSome, it's free and open platform. It's a knowledge base for annotation purposes. And so what VarSome does in the first place is that it aggregates and cross-references fragmented annotation data resources. So on VarSome, you can find currently annotation data from over 70 data resources. So in fact, the interpretation, we can think of it as a puzzle game, right? To get a complete picture, you need to place all the little pieces of evidence in the right positions, right? So with only 2 or 3 pieces, you cannot possibly get the complete picture. So This is the first purpose of VarSome, to bring all these fragmented data resources together in a one place. On top of it, we keep the annotation data coming from all these 70 data resources. We keep them always up to date. Yeah, so you can rest assured we have always the latest annotation data available on VarSome according to the latest scientific discoveries. And on top of it, we implement guidelines for standardization of the interpretation process. So for instance, on VarSome, you can find the automated interpretation of pathogenicity based on ACMG guidelines. I'll cover this part a bit later in my presentation. For now, I'd like to say that we are now also implementing guidelines for somatic interpretation, for example, according to AMP, NCCN, or ASCO. However, VarSome, the free platform, it's not only about data integration and keeping the data always up to date. It's also about you, about VarSome users, right? So on VarSome, we have over 200,000 users using the platform globally, and which explains why, I mean, it's so convenient and platform that it attracts attention, you know, from users all over the world. And so as a consequence of it, there is also a growing number of contributions flowing in from our global community. So VarSome users link publications, link functional studies to variants, classify variants, comment on variant, or start new collaborations. In other words, there is also a unique layer of annotation data, which you can find, you know, obviously only on VarSome. Now, on top of the free platform, we have developed a VarSome Clinical. VarSome Clinical is already a professional paid service, which allows you to process your NGS data starting from FastQ or from VCF. And at the end, you get a clinical report. So basically where Swift Biosciences ends, right? That's where we start. Now, VarSome Clinical is clinically certified. It's certified as an IVD device, certified as an in vitro diagnostic medical device. Our company is also certified with 2 ISO certificates, one for quality management of medical devices, and the other one, 27001, for data security and data privacy. We are obviously also GDPR compliant, and our platform is also HIPAA compliant, which is relevant for our US-based customers. And so with VarSome Clinical, you can generate a clinical report in 4 steps. First, you are supposed to upload your sequencing data, your FastQ files or VCF files. You are supposed to upload it to our servers. So for most of our clients, you know, use our physical server located in Switzerland. Yeah, so that's where you are supposed to upload your sequencing data, to our server in Switzerland. With VarSome Clinical, you can process any kind of NGS data, be it a whole genome, an exome, clinical exome, whole exome, or a specific gene panel. It can be a commercial gene panel, such as gene panels developed by Swift Bio, or it can be even your custom design gene panel. As long as you sequence on Illumina or MGI. And when it comes to the VCF files, there are more possibilities. We can take a VCF file. We can take basically any VCF file as long as it conforms to the standards for VCF files. So that can be a VCF file, you know, from Thermo Fisher, Ion Torrent, FastBio, and so on. Once you upload your sequencing data, you can run the pipeline. VarSome Clinical comes with a wide range of pipelines covering pretty much all the clinical use cases. So for example, We have pipelines, obviously, for germline samples, for somatic samples, for trios, for de novo variants in trios, for carrier risk screening, for couples, a pipeline for copy number variation, structural variation, including a pipeline supporting UMIs, unique molecular identifiers, allowing the detection of low frequency variants typically found in somatic samples. So a wide range of pipelines covering pretty much all the clinical use cases. VarSome Clinical runs on our server in Switzerland, and you access the platform over the internet as if it was a regular website, which means you don't need to maintain your own server. You don't have to worry about data security, about security patches, about data privacy. We take care of all these things, and you just simply upload your data, run the pipeline, and get the clinical report. And as the last step, you can generate a clinical report which can be fully customizable. Basically, you have a complete control over the clinical report. You can set up your own template according to your branding policy, and then you also have control over the annotation data included in the report. So it's a kind of interactive reporting. Here, I'd like to stress out again where the challenge is. Here, the challenge, really lies in the interpretation part of the pipeline. I mean, to do the read alignment and variant calling is relatively simple. The real challenge indeed lies in the interpretation part, and that's where you need to bring as many annotation data resources as possible together, right, to get a complete picture. So this is basically one of the main advantages, of our tools, the interpretation part. However, VarSome Clinical, it's not a replacement for an in-house bioinformatics expertise. In fact, VarSome Clinical is a tool which allows your bioinformatician to work more productively, more systematically, and increase the energy where it is really needed. I mean, for example, for more complex cases. And so over time, VarSome Clinical allows you to increase the diagnostic yield for your patients. Here, excuse me. Here, I just wanted to share very quickly, the list of our clients. So we have clients in pretty much all the European countries, as well as on the global scale. We have a number of customers in the US, in Latin America, as well as in Asia. Now, from the pricing perspective, VarSome Clinical does not have a license. VarSome Clinical charges on a per sample basis. When it comes to the FASTQ files, FASTQ samples, the price depends on the number of megabases in reads. And when it comes to the VCF files, the price depends on the number of variants. There are basically two modes how you can use VarSome Clinical. You can use it as the so-called standalone solutions. Standalone solution is open for any kind of NGS sample, a gene panel, exome, genome, doesn't really matter. And we charge you dynamically based on the usage of the platform on a per sample basis. So this is nothing else than pay as you go model. The other option is the so-called bundled solution. And this is where our partners such as Swift Biosciences come into the picture. The bundled solution is delivered, through a collaboration with our partners such as Swift Bio or, TATAA as our distributor. So the bundled solution contains everything, right? It contains the kit for example, 48, 56G Swift samples bundled with VarSome Clinical for data interpretation and reporting. So this is a form of prepayment where you can prepare, a certain number of analyses for a specific gene panel, for example. And the third option is a local or on-prem installation. We also offer on-prem installations for large-scale sequencing centers or hospitals with a large volume of samples. In the Nordic countries, in Sweden, in Norway, and in Denmark, we are represented by TATAA, who has just become our distributor. TATAA is our joint distributor together with Swift Biosciences, so you can inquiry, for example, for the bundled solution directly to TATAA as our joint distributor. All right, so this has been just a very quick introduction to our company and to our products. Now, I'd like to give you a quick tour through the user interface of our tools. So here first, you can see the free platform, the VarSome knowledge base. So I'd like to actually now share with you the results, annotation data for a particular variant in a BRAF gene. Yeah, so here on a single page, as you can see, this is a very, very long page. I'm not going to go into a great detail, but I just want to show you that on a single page, you get the annotation data from over 70 data resources, always up to date. So you can check that out later. However, I'd like to pause for a while here when it comes to the interpretation based on ACMG guidelines. So this is our main expertise and main focus, the interpretation part of the pipeline. And to do that properly, we first aggregate the fragmented data annotation data resources, and then on top of it we implement guidelines for interpretation of pathogenicity based on ACMG, for example. So for example, in this case here you can see that 6 criteria fired based on the evidence coming from all these data resources. So in other words, our implementation of ACMG guidelines, it's very robust and very solid. It's also very transparent implementation of ACMG guidelines, because every criterion here, you can see that every criterion, it's explained. It's explained why it fired or why it didn't fire. So this is something what allows you to review each individual situation and possibly adjust the final verdict. And third, it's also flexible implementation. It allows you, to specify, for example, the transcript, which may change the final verdict, or you can also specify the supporting strength of the evidence in line with ACMG guidelines. Wherever possible, we consider multiple pieces of evidence. So for example, when it comes to the PP5 criterion, here you can see it considers ClinVar entries, it considers UniProt entries, and also contributions from our global community. So in this particular case, you can see that we consider 3 pieces of evidence. And we do that for every single criterion. Now, on top of the free platform, we have developed VarSome Clinical, which it's a complete solution where you can start directly from your raw sequencing data. With the free platform, you can look up only individual variants, right? But with the free platform, with VarSome Clinical, you can start directly from FastQ or from VCF. So this is a very complex platform. A full demonstration would take over an hour. And so now instead, I'm going to focus on the most important aspects. So first, obviously, you are supposed to upload your sequencing data. You can upload your data over the user interface here, or you can also leverage the API, application programming interface, for a fully automated transfer of your data. So once you upload your data, you can proceed and you can start the pipeline. There are several options, starting from FASTQ, from WISP, PCF, tumor normal pairs. You can perform a trio analysis, carrier risk for couples. You can also run a pipeline for CNVs and structural variation. So for example, let's say we are starting from FASTQ, so we have to also specify whether this is a single sample, whether it's a family trio, couple, or generic multi-sample analysis for extended families or cohort studies. You have to specify whether it's germline or somatic pipeline. Here you should specify the assay, the library preparation assay, such as Swift Biosciences 57G or 56G. And when it comes to the custom gene panels, you need to provide us the BET file with specification of target regions. But in principle, you can process any kind of NGS data, commercial gene panels, custom gene panels, exomes, genomes, as long as you sequence on Illumina or MGI when it comes to FASTQ. You may also optionally specify the ethnicity of your patient. You can select your preferred reference genome, AG 1938. And then on the left side here, for example, you shall specify the sequencing data. You can provide sample IDs, descriptions, and here this is very, very useful. Here you can type in, the patient phenotypes and diseases according to HPO, the human phenotype ontology, such as retinopathy, for example. So you can type in, all the phenotypes and diseases of your patients, and later in the process you can filter your variants based on number of matches with these phenotypes. So this basically allows you to prioritize certain variants over other variants. And then you can already start, the analysis process. For your information, an exome starting from FASTQ takes about half an hour. A whole genome takes about 4 or 5 hours, and gene panels, that's the matter of a few minutes to 20 minutes. So once the analysis is finished, you get the results in the form of the variant table. So for example, here you can see the results for a Swift Biosciences 56G pan-cancer panel with 15 variants. I'll get back to it later, but for now, I'd like to show you first a whole genome, which allows me, to show you, all the advanced filtering capabilities. For example, so here you can see there are only almost 5 million variants it's a whole genome sample. Variants are sorted by pathogenicity according to the ACMG guidelines here. So again, here you can see the ACMG grid. You can see the detailed explanation for each particular criterion, and you can also possibly, modify the verdict by triggering additional criteria according to your specific knowledge for the particular patient. In fact, not all the ACMG criteria can be fully automated. Certain criteria such as PM3, PP4, BP5, these criteria require a patient-specific information, which we don't obviously have. So that's you in those cases who has to take it into consideration and adjust the criteria in line with your best knowledge for the particular case. Then around the variant table here, you can see all the annotation data. So first ACMG, then you can see here the region browser where you can see all the variants within a given gene with the supporting evidence coming from all these public data resources. So for example, for this particular variant here in the first one, here, you can see there are, evidences in UniProt, ClinVar, DBSNP, Caviar, ICGC, Cosmic, Bravo, and so on. And then here you can see the ClinVar entries, Cosmic entries, Caviar data, population frequencies, number of somatic data resources such as ICGC, Cosmic, Bravo, GDC, GTEx, expression profiles from the GTEx consortium, in silico predictions, number of in silico predictions, conservation scores, list of publications linked to the particular variant or gene, and also contributions from our global community. Let's see if there is no. Well, and then on the right side here, on the right side of the screen, you can see another set of annotation data, all the clinically relevant information, all the transcripts, coming from RefSeq and Ensemble, clinically relevant data for genes, for phenotypes and diseases according to the HPO terms, the human phenotype ontology, nearby variants, and so on. Now, let's have a look on the variant table here. So for every single variant here, you can see the classification, the list of ACMG criteria, HGBS term, exon number, mode of inheritance, function. So when it comes to the function, we annotate against all transcripts available, zygosity, frequency, this is the population frequency according to the ethnicity of your patient, allelic balance, the percentage of reads, and coverage, which is the number of reads. You can click on it and it takes you to the JBrowse view where you can see the graphical representation of reads. IGV as a visualization tool, it's also an option here. Now, let's talk for a while about custom classifications. So, for example, here you can see this is a pathogenic variant according to the ACMG guidelines. Yeah, you can review the criteria which fired. However, if you don't agree with the default verdict, you may reclassify the variant internally, only within your account. You may reclassify it as a benign or variant of uncertain significance. Yeah. Or you can set up your fully customized classifications. And again, you can apply them to your variant. So that's why here you can see here all these marks, these tags. These colored tags represent your custom classifications or even the custom ACMG verdict. So you may specify, other criteria, and you can save it here as a manual classification. And once you do that, it becomes a custom classification here in this column. So the most important, thing here is that these custom classifications stay with the variant, yeah, which means that whenever you upload a new sample, you will see here the same classification mark. So this is something what allows you to build over time your private database of classifications. We can even take your manually curated list of variants. I guess you might have it in a spreadsheet, and we can take it and upload it privately to your account with VarSome Clinical for the purposes of variant classification. And the other feature which allows you to increase the diagnostic yield is the sample cross-referencing. So the VarSome Clinical links all your samples together on the variant level. Yeah. So for example, again, this particular variant here, you can see that it has been found in a heterozygous form in 3 samples in your account. Yeah. So this is, again, something what allows you to go back and forth between your past cases and current cases, sharing the same variant, compare the patient phenotypes and diseases, compare allelic balances, and over time, together with custom classifications, it allows you to increase your productivity and, most importantly, the diagnostic yield. So this is all about, increasing, your productivity and your diagnostic yield. Here you can also access number of quality control reports. You can download the BAM file, BCF file. You can download the index BAM file. You can download, for example, the coding coverage report. So coding coverage report, this is an Excel sheet with the coding coverage report, broken down by individual genes, exon numbers, and sequencing that. So here you can basically identify, the regions with a low coverage, or regions which have been skipped for some reason. So this sheet allows you to troubleshoot, sequencing issues or issues with assay design, for example. And then there is a number of QC reports available. Now, I'd like to talk very quickly about filtering. Filtering is also very important. It allows you, to narrow down the list of variants as quickly as possible. So VarSome Clinical comes with two types of filters. The first one you can access on the left side of the screen here. So here you can see the dynamic filters. So with dynamic filters, you can filter your variants based on number of criteria, for example, based on ACMG criteria, based on allele frequency, based on chromosomal position, ClinVar classification, based on zygosity, function, whether it's 5UTR, coding, splicing, intronic variant, gene list. You can set up a gene list basically and use it as a filter for your variants. Call status, you can filter based on the allelic balance or coverage, number of reads, and so on. And so just to give you a very quick example, let's say we want to filter out variants with a low frequency, less than 1%. And on top of it, we want to filter out only pathogenic and likely pathogenic variants. So now we have set up a filter set with two filters. We give it a name, and we can apply it to our sample right away. So also now, please, here you can see actually that filtering works really fast, right? We are working here with a whole genome sample with almost 5 million variants. So here you can see it based on the frequency filter. We have eliminated over 4.5 million variants, and based on pathogenicity, additional 339 variants. You can always, deactivate certain filters, or you can bring them up again. And as you can see, the variants in the variant table update and dynamically. That's why we call them dynamic filters. You can, for example, now include additional filter based on PP5 criterion with a supporting strength of the evidence. So now we have 3 filters within the filter set. We can apply it again, and now there is no pathogenic supporting. There are no variants meeting all 3 criteria, so we can deactivate a certain filter and use only these 2 filters for now. However, that's not all. VarSome Clinical also comes with algorithmic filters. So algorithmic filters are basically a little programs we build specifically for each client, depending on specific needs and workflow for a given client. So these algorithmic filters can be fully customized. For example, here you can see several examples with algorithmic filters. You can perform de novo analysis in trios, compound heterozygous variants, carrier risk for couples, segregation kind of analysis, and so on. Basically, with algorithmic filters, we can filter the variants exactly the way you need. So you just need to tell us how you want to filter your variants, and we develop a specific algorithmic filter for you. So all these filters, the application of these filters, it's all included in the sample price. Remember that VarSome Clinical charges on a per sample basis. Yeah. And then all these sophisticated filters based on gene lists, based on algorithmic filters, and so on, these are all included in the original sample price. Now, just very quickly, the reporting. So once you decide you want to report a particular variant, you select it for export, and then you can proceed, and you can either download the list of variants in an Excel sheet, for example, or you can proceed and you can generate the clinical report. So the clinical report can be fully customized according to your branding policy. You can set up, you can upload your own logo, you can customize the header, you can set up your company details here on institution details. The report can be also translated. I mean, the common elements can be translated, the annotation data, are always in English, obviously, but the common elements of the report can be translated into your language. So basically what you need to do is to set up a template for your report, and then you can use it all over again. Now on the left side here, you can see the list of widgets. In other words, the list of annotation data available for a particular variant. Yeah, so basically, you may drag in a variant information, but first you may want to drag in a content area for your specific comment. And then you can drag in, you know, a gene, clinically relevant information for genes. Then you can drag in, you know, the list of drugs, for example, a list of clinical trials, and at the end, you know, list of publications. Everything is fully editable. So here is the area where you can provide your custom comment for the report. Here you can see the information about the variant, transcript, HVS term, exon number, clinically relevant information for genes, phenotypes, diseases, list of drugs, list of clinical trials, and list of publications. Again, everything is fully editable, so you may remove certain publications, and you can add your own publication, for example, here. So this is the interactive reporting, and then once you are done with it, you can download it as a PDF file or as a doc file, which is basically then the end of the analysis. So as I was saying before, VarSome Clinical, it's a very complex platform which supports a number of pipelines for a wide range of clinical use cases. VarSome Clinical, it's clinically certified. It's certified as an IVD device, and you can process any kind of NGS data, exomes, genomes, or gene panels, either custom built or commercial gene panels designed, for example, by Biosciences. And there are basically two ways how you can pay for the service. You can pay either, we can charge you either, you know, flexibly based on the usage of the platform, or you can prepay a certain number of analyses. This is the so-called bundled solution, which is available, you know, in collaboration with certain partners such as, you know, Swift Biosciences. With that, I think I'll end up my presentation, and I will be very happy to take some questions from the audience. Do we have some questions, Petra?
Thank you, Thomas. Not at the moment. I think we probably forget to mention that there is a chat and a Q&A box here in Zoom. So please use either of those if you have any questions. We do have some time. And then, as Thomas mentioned, it's a complicated platform, and a proper introduction would take more than 1 hour. So if this is something you would like to do, we can of course schedule that with you on a later date.
Indeed, yeah, I would be very happy to do that on a case-by-case basis. Also, for your information, here we have just published here the list of learning resources available on varsome.com/library. So, for example, here you can learn about, you know, the technical details of ACMG-based interpretation. Here you can learn how you can increase the diagnostic yield with VarSome Clinical, and basically you can basically educate yourself about our platform here. You can also watch a learning VarSome Clinical series available on YouTube, which basically guides you through the whole analysis process, starting with data upload all the way to the clinical report generation.
I do see a question here. Could you please provide an estimate price of running full analysis of one whole genome sequencing?
Mm-hmm. Yes, and so It really depends. You know, if you, I assume you would be starting from FASTQ. So when it comes to the FASTQ, the price depends on the number of megabases in reads. In other words, it really depends on the sequencing depth. Yeah. Certain laboratories sequence deeper, other laboratories less deeper. So it really depends, you know, how deep you sequence your sample. But to give you a ballpark number, it can be around, yeah, around 250 perhaps, but it really depends on the sequencing depth.
And that would be euros, right?
Mm-hmm. Mm-hmm.
Yeah. Any more questions?
I see some question here. See, I saw a question, but I can't access the control panel. How is that possible? Here. Okay. How is the question about the price estimate for a whole genome? Okay. All right. If there are no questions, I'd like to thank you all for taking time for joining us today for our joint presentation with Gerardas from Swift Bio and with Petra from TATAA, our joint distributor. I hope it was a productively spent time, and thank you for your attention. Any remarks, Petra, Gerardas?
No, thank you, Thomas and Gerardas.
Oh, sorry, Petra.
Yeah, no worries. And thank you, everybody, for participating.
Right, thank you very much.
If you got any questions, please follow up with Petra. Petra will follow up with you, and we would be happy to provide the answers. Thank you.
Yes.
All right, thank you all.
Thank you
I'm
very
going
much.
to close the meeting now. Bye-bye.
Bye.